Welcome! In this video tutorial, I will briefly show you how to annotate cells in Advanced Cell Classifier. Annotating cells is essential for supervised learning, during which expert knowledge is provided to the computer to enable automatic data analysis. I'm going to describe how to use the traditional and the more advanced annotation methods and, additionally, how to explore your data by using a weakly supervised method. First of all, the user may define classes of interest by simply using the Create Class button. Then a window appears where the name, type, and icon of the class can be specified. Type in the name, for example, normal cells. The next parameter is the type. The user can choose between two types of classes, normal and child. For basic purposes, you can use the normal class. If you want to define a hierarchy in your annotations, you can define child classes for a normal class. Finally, choose an icon for the class, in our case, the currently selected cell. Repeat this process to define new classes as many times as you wish. For instance, here we are going to define the class of rounded up cells as well. The easiest way to annotate cells is to use the manual annotation modality. In this case, you can use the image selector window to browse through your images. Then you can assign the cell to a class by first clicking on the cell and then on the icon of the appropriate class. After assignment, the highlight automatically jumps to the next cell on the image. Manual annotation can result in biased data when the classified cells are not sampled uniformly. To overcome this, Advanced Cell Classifier offers a random annotation option. By clicking on the Random Annotation button, the program will jump to a randomly selected cell you may classify next. It is not required to classify the proposed cell. You can override the random selection anytime. When you want to turn off random annotation, simply click again on its specific button. The following more sophisticated annotation methods of Advanced Cell Classifier automatically search your data in intelligent ways. This requires fast access to the features. To speed up the process, we provide the possibility of loading the entire or a subset of the data into the memory. These data are referred to as the sampling pool. You can create a sampling pool by using the Generate Sampling Pool button. In the option window, you can specify the ratio of the samples that you want to load. For example, set the ratio to 0.5 if you want to load half of your data. Advanced Cell Classifier is equipped with active learning methods to improve the quality of the training set and then the accuracy of the classification by simply employing the computer to show cells that are difficult to automatically classify with the current sets of annotated cells. In practice, when active learning is on, several difficult cells will be shown to the user. Different active learning methods are implemented. However, committee members with the size of committee 3 typically provide good results. Now, by pressing the Start Active Learning button, cell selection will jump to an automatically selected cell that may improve classification the most. To turn off active learning, simply press this button again. Besides the presented annotation methods, Advanced Cell Classifier contains tools that help you explore your data. For instance, by simply pressing the Find Similar Cells button, you can automatically find the cells that are the most similar to the currently selected one. This will show you a window with the discovered cells. You can then easily annotate these cells. Select one cell shown in the window, press the Jump to Cell button and click on the desired class. The last functionality presented in this video is the Phenotype Finder. It offers a way to automatically identify new interesting cell types. In fact, Phenotype Finder is the nickname of a weekly supervised machine learning module we implemented with the aim of finding interesting object classes automatically by analyzing the space of the non-annotated cells. Phenotype Finder works in a multi-dimensional feature space, represented here as a two-dimensional one for the sake of clarity. Starting from the annotated cells, green, blue, and yellow regions in the figure, it defines the region of the non-annotated cells, red region. Then, by analyzing the cells in the red region, it builds a hierarchical tree with subgroups of cells with similar phenotypes. This may help the user find new classes of interest. This will show you a window with a binary tree where you can explore the structure of non-annotated cells. By clicking on a node in the tree, it will open two children representing a possible binary classification for the samples in the parent node. The green rectangular button in the bottom left corner shows all cells that belong to the current subtree. The red button allows the user to directly define a new class and insert all the underlying cells. You have now been given an insight into Advanced Cell Classifier's annotation methods. To learn more about the different parameters, please read the help documentation file provided with the software. Thank you for your attention.